Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Artesunate is usually thought of as an anti-malarial drug, but cancer researchers have been asking a very different question. Can this drug help push cancer cells into ferroptosis, an iron-driven form of cell death? This is where the story gets very fascinating because artesunate does not appear to act through just one pathway. It may react with iron to generate free radicals. It may increase the labile iron pool. It may break down ferritin, the cell's iron storage system. It may weaken glutathione, glutathione peroxidase, FSP1, antioxidant defense systems. It may help drive lipid peroxidation into PUFA-rich membranes, the final destructive step of ferroptosis. So this is not just an iron-activated ROS story. This may be a 7-way ferroptosis attack. Today, we're going to break down how artesunate may attack cancer survival through 3 major mechanisms: iron dysregulation, antioxidant collapse, and lipid peroxidation. So without further ado, let's get into it. So how does artemisinin artesunate affect cancer cells specifically through iron, ferritinophagy, and ferroptosis? So let's start with looking at artesunate through the lens of artemisinin, its natural counterpart. And this paper was published in August of 2024 titled Artemisinin and its Derivatives as Potential Anti-Cancer Agents. And it says here, artemisinin is a natural sesquiterpene lactone obtained from the traditional Chinese medicinal herb Artemisia annua. Artemisinin and its derivatives share an unusual endoperoxide bridge that are extensively used for malarial treatment worldwide. In addition to antimalarial activities, artemisinin and its derivatives have been reported to exhibit promising anti-cancer effects in recent decades. In this review, we focused on the research progress of artemisinin and its derivatives with potential anti-cancer activities, the pharmacological effects, potential mechanisms, and clinical trials in cancer therapy of artemisinin and its derivatives were discussed. So I show this only so we're on the same page of what we're actually talking about with artesunate. So there's the actual plant, then there's the extraction of artemisinin, and then there's chemical changes to make dihydroartemisinin, and then further chemical changes to make the pharmaceutical drug artesunate. And we're going to be focusing mostly on artesunate today. And I pulled this paper from October 2025 titled The Anti-Tumor Mechanism of Artesunate. And I wanted to show this because it had some really interesting graphics that went along with it. First of all, it's been studied in multiple cancers, and it's also been shown to have multiple mechanisms of action. And I think that's probably illustrated even just as powerfully on this particular graphic that was also part of this paper. And what you're seeing here is there's inhibition of tumor cell proliferation, invasion of metastases, induction of apoptosis and autophagy, inhibition of tumor angiogenesis, overcoming drug resistance in cancer cells, affecting the tumor microenvironment, and modulating the immune system. And what I did is I took these very common metabolic pathways and I put them on a graphic that really shows how many of these, if not all of these, in one way or another, affect the metabolic reprogramming of cancer, whether it be through the upregulation of GLUT transporters and glycolysis through the Warburg effect, or through the upregulation of glutamine transporters and glutaminolysis through mitochondrial substrate level phosphorylation, or the making of energy or the fermentation of glutamine. So artesunate is going to have effects on many, many systems, including glucose and glutamine. But how does it relate to ferroptosis? That's what this video is about. So I'm going to start with talking about kind of all the major mechanisms that have been discovered regarding artesunate's ability to activate or enhance ferroptosis. And I'm going to start with the most classical pathway, which is its free radical pathway regarding the endoperoxide bridge that it has. So artesunate's structure is unique. It has this endoperoxide bridge, which is a key reactive feature. When it reacts with iron, it cleaves the bridge and generates reactive oxygen species in the form of different types of ROS. And those ROS then can cause oxidative stress within the cell. So one mechanism of how artesunate works, and probably the classical mechanism of how artesunate works, is the generation of reactive oxygen species in interacting with iron. But that is not all. The next paper I'm going to talk about was published in July of 2022, and it's titled Intracellular Ferritin heavy chains plays the key role in artesunate-induced ferroptosis in ovarian serous carcinoma cells. And it says here in the abstract, artesunate, an antimalarial drug, induces ferroptosis, but the mechanism is still unclear. In the present study, we investigated how artesunate induces ferroptosis in ovarian serous carcinoma. Experiments were performed using the ovarian serous carcinoma cell lines CAOV3 and SKOV3IP1. And the sensitivity of CAOV3 to artesunate was higher than that of SKOV3IP1. Inhibited artesunate-induced intracellular lipid peroxidation and cell death. However, unlike class 1 ferroptosis inducer Aristin, artesunate had no effect on the intracellular glutathione SH levels. We found that artesunate-induced changes in lysosomal iron were parallel to the induction of ferroptosis. Therefore, ferritin, which oxidizes and binds intracellular iron, may have an inhibitory effect on ferroptosis. Knockdown of nuclear coactivator 4, a key molecule in ferritinophagy, ferritin-specific autophagy, suppressed artesunate-induced cell death. Knockdown of ferritin heavy chain by siRNA greatly enhanced the sensitivity to artesunate, and overexpression of ferritin heavy chain greatly reduced the sensitivity of ovarian cancer cell lines to artesunate. These results can explain the differential sensitivity of the CAOV3 and the SKOV3IP1 to artesunate. In conclusion, enhancement of ferritinophagy is an important step involved in the mechanism of artesunate-induced ferroptosis, and ferritin heavy chain levels may contribute to the regulation of sensitivity in artesunate-induced ferroptosis in ovarian serous carcinoma cells. And so that begs the question, really, what is ferritinophagy? Well, as it mentioned here, ferritinophagy is the autophagy specifically of the protein ferritin. And ferritin is an iron-binding protein that helps protect against ferroptosis. So what this paper is actually saying is that artesunate is enhancing ferritinophagy, which allows for more free iron to be available to the cell. That is contributing to what they call the labile iron pool. And then that labile iron pool can either react with artesunates that's already there, its endoperoxide bridge, and create further free radicals and lipid peroxidation, or it can have other effects on the cell, which we will talk about. And this is just another paper that's talking about how artesunate induces ER-driven ROS mediated cell death by disrupting labile iron pool and iron redistribution in liver cancer or hepatocellular carcinoma. And basically what it's doing is it's changing the lysosomal pH, and that allows for free iron to escape and enter the labile iron pool, which then is now a trap basically set by artesunate. But that's not all. This paper was published fairly recently in November 2025. It's titled Artesunate Induces Ferroptosis in Gastric Cancer by targeting this TFRC HSPA9 axis for iron homeostasis regulation. And it says here in the abstract here, we identify artesunate, an antimalarial drug, as a potent inducer of ferroptosis in gastric cancer cells and reveal the transferrin receptor, TFRC, as a key mediator in this process. Notably, our study is the first to demonstrate the interaction between artesunate and TFRC through molecular docking and surface plasmon resonance experiments, highlighting a novel mechanism by which artesunate stabilizes TFRC by inhibiting its lysosomal degradation. This stabilization is regulated via the heat shock protein HSP9A, another previously unreported interaction. Disrupting the TFRC HSPA9 interaction facilitates iron accumulation and lipid peroxidation, hallmark features of ferroptosis leading to significant cancer cell death. So now we've explored how there is direct reactive oxygen species that are created by artesunate, the classical pathway. Then we know that it actually breaks down the iron-binding protein ferritin through ferritinophagy, so there's more iron available. Now what we're discussing is how there is an enhancement of iron uptake into cancer cells, and the next mechanisms will discuss how artesunate can interact with antioxidant systems inside the cells to further enhance this process. This paper was published in January of 2026. Artesunate nanoliposomes in hepatocellular carcinoma, mechanistic ferroptosis induction and translational opportunities. And we're going to go through this paper. This is the only paper we're actually going to go through in detail because it is now taking all of the major understood mechanisms, and there may be more in the future, who knows, but it's taking all of the known mechanisms of how ferroptosis works up until this point, and it breaks them down. So pathways of ferroptosis induction in hepatocellular carcinoma cells by artesunate. In September 2025, the latest study by Wang's team demonstrated that artesunate not only induces lipid peroxidation by increasing intracellular iron concentrations and ROS accumulation, but directly binds to transferrin receptor TFRC, that was the paper we looked at, blocks its interaction with heat shock protein HSP9, therefore inhibiting the lysosomal degradation pathway of TFRC, promoting iron uptake and exacerbating the ferroptosis process. Due to their high invasiveness, hepatocellular carcinoma or liver cancer cells exhibit rapid proliferation and significantly increased demand for iron compared to normal hepatocytes or liver cells, a phenomenon directly associated with the marked upregulation of transferrin receptor expression on the cell surface. During hepatocellular carcinoma progression, iron metabolism-related pathways exert precise regulatory effects on the state of cancer cells. Ferroptosis and HCC is driven by iron overload, lipid peroxidation or oxidative stress with lipids, and impaired antioxidant defenses, XC, glutathione, glutathione peroxidase, FSP1, CoQ10. Artesunate's endoperoxide bridge reacts with labile iron to generate ROS, while lipid nanoliposomes increase hepatic delivery and lysosomal trafficking, functionally converging GPX depletion, or glutathione peroxidase 4 depletion, lipid peroxidation, and ferroptotic death. That's kind of the overall gestalt. Regulation of ferroptosis by iron metabolism pathways. Iron in the organism mainly exists in two forms: ferrous iron, Fe2+, and ferric iron, Fe3+. Excessive Fe2+, or ferrous iron, can generate metabolically toxic ROS through catalytic reactions, which oxidize the lipid components of cell membranes and initiate the ferroptosis program. Intracellular 3+, iron 3+, must be reduced to Fe2+ under mediation of divalent metal transporters and to participate in subsequent metabolic processes, where excess iron beyond cellular demand is stored in ferritin, thereby maintaining a dynamic balance of intracellular iron storage. In hepatocellular carcinoma, abnormalities in the expression levels or functions of key iron regulatory molecules such as transferrin and hepcidin may directly affect cellular iron uptake, transport, and utilization, therefore participating in ferroptosis-related tumor regulatory mechanisms. As a potential ferroptosis inducer, artesunate can promote lysosome-mediated ferritin degradation to induce the release of stored iron as free iron ions, thereby significantly increasing intracellular iron concentration, the dangerous type of iron concentration, the toxic type of iron concentration to cells. Meanwhile, it can inhibit functions of iron metabolism, regulatory proteins, including transferrin receptor, TFRC, and heat shock protein, HSPA9, disrupting cellular iron homeostasis at multiple levels. Mediated by free iron ions, the endoperoxide bridge structure of artesunate cleaves to generate ROS, triggering cascading lipid peroxidation reactions that cause sustained oxidative damage to membrane lipid components. Ultimately, this activates the ferroptosis pathway, achieving the inhibition of hepatocellular carcinoma cell growth. And I feel like an infomercial announcer saying there's more, because there is more. Regulation of ferroptosis by artesunate via the XC glutathione peroxidase pathway. The system XC glutathione peroxidase 4 pathway acts as a critical antioxidant defense against ferroptosis, and its functional impairment directly dictates the sensitivity of hepatocellular carcinoma and many other cancers to ferroptosis. Dysregulation of system XC has been documented in various liver disorders, including liver injury, non-alcoholic steatohepatitis, NASH, now called MASH, also metabolic associated steatohepatitis, liver fibrosis, cirrhosis, and hepatocellular carcinoma. System XC is a transmembrane transporter complex consisting of two subunits along the light chain, including XCT, solute carrier family S member 11 SLC 7811, is primarily responsible for its transport activity. This transporter mediates the antiport of extracellular cysteine and intracellular glutamate. Cysteine serves as a key precursor for glutathione synthesis. Adequate intracellular cysteine levels support sustained high glutathione concentrations, and glutathione cooperates with glutathione peroxidase 4 to scavenge excess lipid peroxides, thereby preventing lipid peroxidation-induced ferroptosis. Glutathione peroxidase 4 is well-characterized core inhibitor of ferroptosis that directly reduces lipid hydroperoxides to non-toxic lipid alcohols. A coenzyme for glutathione peroxidase 4, GPX4, inhibition of de novo glutathione synthesis results in glutathione peroxidase 4 inactivation and subsequent induction of ferroptosis. Artesunate can directly suppress the transport activity of system XC, reduce cysteine uptake in hepatocellular carcinoma cells or HCC cells, and block glutathione synthesis at the source. Using radioisotope labeling experiments, Wang et al. Demonstrated that the treatment of Hep G2 cells with artesunate at 50 micromoles per liter for 24 hours decreased cysteine uptake by 42.3% compared to the control group, and this inhibitory effect was dose dependent. Mechanistically, artesunate binds to the active site of SLC711, interfering with its specific interaction with cysteine and thus inhibiting transport function. Further molecular docking experiments revealed that the endoperoxide bridge of artesunate forms hydrogen bonds with cysteine residues at the SLC711 binding site, stabilizing the drug protein complex and exerting sustained inhibition of the XC system. This is new. I had no idea until I read this that artesunate actually directly inhibited SLC711, also known as in some publications the Achilles heel of the tumor, is a major node of inhibition. I was not aware that it directly inhibited. I was aware that it affected expression level, which the researchers also talk about in this next piece. Regulation of ferroptosis by artesunate via the p53 SLC711 ALOX12 pathway. The p53 SLC711 12 lipooxygenase or ALOX12 pathway serves as a critical signaling axis in ferroptosis regulation, precisely modulating the ferroptosis sensitivity of hepatocellular carcinoma HCC cells through the upstream regulation intermediate hub downstream effort mode. P53 is a classic tumor suppressor gene involved in regulating cell proliferation, apoptosis, differentiation, and metabolism. One of its downstream targets is SLC711, a protein that mediates extracellular cysteine uptake to support glutathione synthesis. Mechanistically, SLC711 belongs to a genes associated with apoptosis and redox regulation. P53 can directly bind to the promoter of the SLC711 gene and inhibit its transcription. Additionally, P53 indirectly activates ALOX12 by suppressing SLC711 expression and reducing cysteine uptake. Under ROS stress, this leads to ALOX12-dependent ferroptosis. However, p53 activation exerts no significant effects on glutathione peroxidase 4 function, indicating that p53 induces ferroptosis independently of glutathione peroxidase 4. Thus, the p53 SLC7A11 ALOX pathway is considered or recognized as another critical regulatory pathway for ferroptosis. Artesunate can activate the p53 pathway by inducing DNA damage, enhancing the transcriptional repression of SLC7A11 by p53. A study by Zheng et al. Showed that treatment with SMMC7221 HCC or hepatocellular carcinoma cells with artesunate at 50 micromoles for 24 hours increased the expression levels of DNA damage marker by 2.8-fold and the phosphorylation of p53 at serine 56 by 3.2-fold, demonstrating the DNA damage trigger p53 activation. Following p53 activation, artesunate can further reduce cysteine uptake and block glutathione synthesis by downregulating the expression and activity of SLC7A11. Relevant experimental studies have indicated that after artesunate treatment, cysteine uptake by HCC cells decreased by 52%. Intracellular cysteine levels dropped by 48% compared to the control group, resulting in its insufficient substrates for glutathione synthesis. With the reduction in glutathione levels and accumulation of free iron, artesunate accelerates the production of lipid peroxides by upregulating expression and activity of ALOX12, ultimately triggering ferroptosis. And believe me, there is a picture that goes along with this that makes a lot of this come together. Regulation of ferroptosis by artesunate via the NRF2 and ATF4 pathways. But wait, there's more. Nuclear factor erythroid 2 factor 2, NRF2, the main antioxidant response element, and activating transcription factor 4, ATF4, are core signaling pathways for the cell to cope with oxidative stress and metabolic pressure. By regulating the expression downstream target genes, both pathways collectively maintain intracellular redox homeostasis, the balance of antioxidants to oxidants, and amino acid metabolic balance. During the regulation of ferroptosis and HCC, the NRF2 and ATF4 pathways exhibit crosstalk and synergistic activation. ATF4 can directly bind to the promoter region of NRF2 and promote its transcriptional activity, while NRF2 can indirectly activate the ATF4 pathway by regulating the activity of upstream signaling molecules of ATF4. This interaction forms an NRF2 ATF4 SLC7011 positive feedback loop, which significantly enhances the resistance of ACC to ferroptosis. Artesunate can reduce NRF2 transcriptional activity by inhibiting the dissociation of NRF2 from KEAP1 and blocking its nuclear translocation. Artesunate can significantly inhibit the nuclear translocation of NRF2 in liver cancer cells. In addition, artesunate can block ATF4 interaction by suppressing the PERK-E IF2A ATF4 signaling axis, thereby interfering with the metabolic adaptation of HCC or hepatocellular carcinoma cells. Artesunate has been shown to induce ferroptosis in hepatocellular carcinoma ACC by promoting iron accumulation and lipid peroxidation. It can suppress this antioxidant stress response pathway, thereby the antioxidant defense mediated by ATF4 and NRF2, two key regulators that protect hepatocellular carcinoma cells from ferroptosis. The combined inhibition of these pathways amplifies lipid peroxidation and ferroptosis sensitivity, ultimately enhancing the anti-HCC effect of artesunate. But that's not all. Regulation of ferroptosis by artesunate via the NADPH FSP CoQ pathway. In addition to the classical XC, GSH, GPX4 pathway, the NADPH ferroptosis suppression protein 1, FSP1, coenzyme Q, CoQ10 pathway as a newly formed identified core ferroptosis resistant pathway in recent years, protects cells from lipid peroxidation damage through a glutathione independent mechanism, and antioxidant mechanism plays a key role in the survival of drug resistance of HCC or hepatocellular carcinoma cells. FSP1 is the core effector molecule of this pathway, which is localized to the cell membrane and exhibits NADPH dependent CoQ10 reductase activity. FSP1 can reduce oxidized CoQ10 to reduce CoQ10 by consuming NADPH. As a potent lipid antioxidant, CoQ10 hydroquinone can directly scavenge lipid-free radicals and inhibit the initiation and propagation of lipid peroxidation. In hepatocellular carcinoma, FSP1 is often highly expressed and enhancing the activity of the NADPH FSP1 CoQ10 pathways, which helps tumors escape ferroptosis. Artesunate can rapidly deplete intracellular NADPH in hepatocellular carcinoma cells by generating ROS and activating NADPH oxidase, thereby cutting off the supply of the NADPH FSP1 CoQ10 pathway. What these authors are actually forgetting is that by inhibiting glucose and glutamine, you are cutting off the vast majority of NADPH through the PPP and other related pathways that generate NADPH. So that is probably a major mechanism missed by these authors of why the NADPH is depleted by artesunate. Going back, on the basis of NADPH depletion, artesunate can also directly bind to FSP1, inhibit its CoQ10 reductase activity, and block the conversion of CoQ10Q to CoQ10QH2. With the inhibition of FSP1 activity, reduction of CoQ10QH2 content, artesunate further promotes the oxidative damage of CoQ10Q, disrupts the antioxidant cycle of CoQ10, and accelerates the accumulation of lipid peroxidation by targeting the NADPH FSP1 CoQ10 pathway at multiple steps. Artesunate ultimately induces ferroptosis in cancer cells. And this is the graphic that goes along with this, which I actually made a better graphic that really illustrates the 7-fold decrease or increased in ferroptosis sensitivity. So it's talking about the transferrin protein and basically tricking the cell to bring in even more iron than it normally does. It is going to directly inhibit this system XC, which is the SLC711 antiporter, which brings in cysteine and helps create glutathione. It also downregulates the expression of XC. It also downregulates the expression of glutathione peroxidase 4 via NRF2 and ATF4. And it downregulates again the expression of the SLC711. And it effectively, through a variety of mechanisms, including this FSP1 CoQ10 NADPH system, enhances lipid peroxidation and ferroptosis. And I hope this is not getting old at this point, but this molecule is so unbelievable. But that's not all. Dihydroartemisinin, a derivative of artemisinin and the active metabolized component of artesunate, induces ferroptosis in HCC, again, hepatocellular carcinoma, by promoting the formation of this PEB-P1-15LO. This was published in December of 2021. And it says here, relevant researchers have recognized the role of inducing ferroptosis in the treatment of the tumor. The latest findings indicate that the PEB-P1-15LO can play an essential role in the process of cell death. However, its role in regulating ferroptosis in hepatocellular carcinoma, simplified by HCC, remains unclear. The previous research of our team has proved that DHA, dihydro, can induce ferroptosis of hepatic stellate cells. In this study, we found that DHA could also induce ferroptosis in hepatocellular carcinoma cells. Interestingly, DHA induced ferroptosis by promoting the formation of this PEB-P1-15LO and promoting cell membrane lipid peroxidation. In addition, we found that DHA had no obvious regulatory effects on 15LO, but could promote this PEB-P1 protein expression. Importantly, we discovered that upregulation of PEB-P1 induced by DHA was related to the inhibition of ubiquination downregulation. In vivo experiments have obtained consistent results that DHA can inhibit tumor growth and affect expression of ferroptosis markers in tumor tissues, which would be partially offset by the interference with PEB-P1. So what does this PEB-P1 actually do? So what it does is it works on a completely different system, these LOX, these lipoxygenase enzymes. And these lipoxygenase enzymes will further oxidize membrane lipids, leading to another way that ferroptosis can be enhanced. It says here, lipoxygenases are non-heme, iron-containing enzymatic protein effectors, an important enzyme system that mediates the formation of ferroptosis peroxides. Free polyunsaturated fatty acids are preferred substrates for LOX, and LOX knockout can alleviate aristin-induced ferroptosis damage. LOXes involved in mediating lipid peroxides are mainly 15LO. Recently, it's been found that 15LO oxidizes free polyunsaturated fatty acids, PUFA, with the help of this PEB-P1 to form this PEPB1-15LO complex. The allosteric regulation of LOXes can then initiate the downstream phospholipase A2 related oxidation pathway of specific polyunsaturated fatty acids. 15LO is a novel partner of PEB-P1, and the complex formed by the two can allosterically activate LOXes to initiate the ferroptosis process. Dihydroartemisinin, a derivative of artemisinin, plays a crucial role in the antiviral and antibacterial activities in addition to its antimalarial activities. In addition, the anti-tumor effect of DHA has become a focus of research. At present, a large number of studies have shown that DHA has inhibitory effects on lung cancer, prostate cancer, epithelial ovarian cancer. The mechanism researchers is mainly concentrated in DHA-induced autophagy, cell cycle blocking, and apoptosis, which in turn leads to restricted tumor cell proliferation. Previous research have confirmed that DHA can also result in iron homeostasis imbalance in cancer cells, which can lead to ferroptosis. In addition, previous studies by our team have confirmed that DHA can induce ferroptosis of hepatic stellate cells and reverse the development of hepatic fibrosis. Here we discuss whether DHA can induce HCC and exert antitumor effects by regulating the formation of this PEB-P1-15OL complex. So again, going back to the graphic, it's artesunate or dihydroartemisinin is enhancing this PEB-1-15OL complex, which is allowing for membrane lipids to be further oxidized in a completely different way, further enhancing ferroptosis, which led me to take all of this information and create this graphic of 7 distinct ways that artesunate enhances ferroptosis in tumor cells. It enhances iron import through this TFRC mediated iron uptake. It participates in Fenton chemistry through the endoperoxide bridge. It enhances ferritinophagy and ferritin breakdown, which allows for more iron to be available. It has inhibitory effects through various mechanisms on glutathione peroxidase 4 and other antioxidant systems. It has direct blockade of SLC7811 system XC, and it decreases the expression of that enzyme as well. In multiple steps, can inhibit this FSP1, NADPH, CoQ10 antioxidant system, and it can, lastly, it can work through this lipooxygenase PEB-P1-15LO complex to further lead to lipid peroxidation, which ultimately ends up in a seven-way attack, at least seven-way attack of how artesunate, an artemisinin derivative, enhances ferroptosis in cancer. If you liked to see breakdowns like this, please like, share, hype, subscribe, and until next time.